Now let's talk about pathogenesis of diabetes mellitus. So let's go through the type 1 diabetes. So we already know that type 1 diabetes, it's an autoimmune disease. It's an autoimmune disease which leads to the destruction of beta cells of islets. So ultimately the absolute insulin deficiency, thus causing features of hyperglycemia. That's called type 1 diabetes. So we have read up to this point. So basically, type 1 diabetes is caused by the combination of genetic susceptibility and the environmental factor. So type 1 diabetes is caused by the combinations of genetic and environmental factor. If we talk about the genetic susceptibility, those who have the HLA locus DQ3, it's DR3 or DR4, have high chance, which the thing I'm talking about is about MSC gene. So genetic susceptibility increases of MSC gene type 2 or class 2 HLA locus. So those who have DR3 or DR4 present in HLA locus have high risk of having diabetes mellitus type 1. The research data was the 90 to 95% of the whites who have type 1 diabetes have either DR3 or DRQ4 haplotypes, compared to 40% of normal subjects. So compared to the normal one, the cases of type 1 diabetes, when analyzed, those in the whites, 90 to 95% of the cases, those who have type 1 diabetes have either DR3 or DR4 haplotypes in them in the HLA locus. Similarly, if they have DQ8 haplotype in them along with the DR3 or DR4, the risk of inheritance increases quite significantly. So if they have DQ8 variations is present in them, the risk of inheritance increases quite significantly. So there are, for the genetic susceptibility till now, that MSC gene, HLA locus, DR3 and DR4, DQA if present, high risk of inheritance. That's it. If we talk about non-MSC gene, then the genes involved in them are non-MSC polymorphism associated with the disease are CTLA4 genes, PTP and 22. So these gene polymorphism can be associated with the diabetes mellitus. Along with it, there is, if mutations in the AIRE, autoimmune regulatory genes, which encodes an immune regulator. So basically it is an immune regulator. If mutation occur in it, then also the type 1 diabetes may occur. So up to now we are talking about type 1 diabetes pathogenesis, combination of genetic susceptibility and environmental factors. We'll talk in few seconds. So the MSC gene involved is highly be found. DR3 and DR4 90 to 95 percentage were there compared to the 40% of the normal subjects. Non-MSC gene CTLA4 and PTP-N22 polymorphism is involved, and AIRE mutations. So let's talk about environmental factor. If environmental factors is considered, if we compare it with the type 2 diabetes, the type 2 diabetes have more genetic association as well as environmental association. That means environment and genetic both have greater role to play in the type 2 diabetes. That's why type 2 diabetes percentage is 90% of the cases mainly are of type 2 diabetes. So environmental factor, the thing is found is that viral agents have been found to be the potential trigger of the autoimmune attack. So there is a postulated hypothesis that through the molecular mimicry, they bring the autoimmune attack. That's the postulated hypothesis. So in this, that virus produce a protein that elicit or express the host response through cross reactivity, which we are telling it as a molecular mimicry pathway. This is the environmental factors associated with the type 1 diabetes. Some of the virus that have been associated with the type 1 diabetes are mumps virus, cytomegalovirus, rubella are there. So these are the viruses which are associated with the environmental factors. So to summarize or conclude about the pathogenesis causing type 1 diabetes, these genetic susceptibility and environmental factor make a favorable environment so that autoimmune process gets activated, which will ultimately leads to beta cell destruction, and thus absolute deficiency of insulin will be there. Also there are stages of the type 1 diabetes which we'll be talking now, but before that, how beta cells gets destroyed, let's talk about that mechanism of beta cell destruction. So talking about, we have already knew that it's an autoimmune process. So fundamental abnormality that will be present is failure of T cell self tolerance. So fundamental abnormality is failure of T cell self tolerance. This will be present. So how they happens is there are multiple mechanisms that gets activated. We already know about this in previous chapter, that CD4 T helper cells get activated, then cytokines are released. Cytokines like high interferon gamma and TNF, these factors get released, then macrophage gets activated, thus killing the beta cells. Next is CD8, which is cytotoxic in nature. Cytotoxic T cells are there which directly kill beta cell. Next are there is autoantibodies as well against insulin can be there, and against enzymes like glutamic acid decarboxylase. So autoantibodies against this enzyme or insulin like this phenomena also occur, which can leads to beta cell destruction. So this is the basic mechanisms of beta cell destruction that can occur in type 1 diabetes. Now after that, let's talk that the stages that can occur in type 1 diabetes. So basically, when a patient are presented to us with the symptoms that's been already been laid, because that symptomatic effect that is seen in patients is already in stage 3 of type 1 diabetes. So let's go through the figure. Let's suppose this is the I have already figure in here, sorry. So let's suppose this is the time and this is the functional mass. So if let's suppose a patient is presented with the symptoms, for presenting the symptoms there will be the very less insulin. That means only 90% 10% of the functional mass is remained. In that case symptoms will be seen, that will be stage 3. So in stage 1, so autoimmunity, that is autoantibodies will be present, but glycemia, if we check about it, it will be normal and no symptoms will be seen. That's the stage 1. In stage 2, this glycemia can be seen and the beta cell starts to deteriorate or gets killed. So when 90% of the cells gets killed, then the symptoms starts to appear. This is the regarding the stages of type 1 diabetes. Now at last let's compare some points with the type 2 diabetes and we'll move to pathogenesis of type 2 diabetes. So clinically, type 1 diabetes majorly occur in childhood, but it can also occur in the later stages of the life. Usually or more generally it occur in the adults. So this is the basic. So in type 1 diabetes there will be progressive depletion of the insulin level. We also already talk about the stages involved in there, whereas there will be blood insulin will be normal, or also due to compensation activity can increase, but obviously insulin resistance here is there. So type 2 diabetes. So other thing main involved is genetics. MSC class type 2 gene is involved in them, whereas no HLA linkage is found in type 2 diabetes. So pathogenesis wise, we know that dysfunction of T cell selection and regulation leading to the breakdown in self tolerance to islet autoantigens. So one last thing is insulitis can be seen in there. That means inside the beta cells inflammatory infiltrates gets deposited. That's the features, whereas amyloidosis can be seen in type 2 diabetes.